Welcome to the IonOpticks product demonstration. My name is Jarrod Sandow and I'm one of the founders and head of product development at IonOpticks. Thanks for joining us to hear how our chromatography columns can improve your research. Here at IonOpticks, we truly believe that your data is only as good as your chromatography. You can have the most advanced mass spectrometer in your lab, maybe even several, but without high performance and robust separation of your experimental samples, you're really not able to unlock the full potential of these amazing pieces of equipment. Now, Bruker and Thermo, they understand this, which is why when they wanted to produce data for their brochures, for application notes, they turned to IonOpticks Aurora Series columns to achieve this and really demonstrate the power of their new flagship mass spectrometers. So why are we the trusted choice in chromatography? Our Aurora columns feature several key technological advances. The first is a really refined manufacturing technique that allows us to pack the column bed directly into the emitter. Now, this eliminates all post-column dead volume and gives you amazing peak capacity. The second is our unique nano-zero union. Now, this can only be found on Aurora Series columns. And this is a true zero dead volume union that maximizes separation potential while also being plug and play, and it has amazing pressure holding capacity. It can hold up to 1200 bar back pressure. Combined, these features allow our Aurora Series columns to outperform any on the market. When we did a comparison of our 25 centimeter Aurora Series column to other competitors' 50 centimeters packed bed columns, either with or without an integrated emitter tip on these competitors' columns, what we found was that we were able to achieve greater than 20% better performance in protein identifications. Now, this means that our columns are not only highly sensitive, they have better throughput because they're shorter and more efficient, but you also get amazingly deep proteome coverage. So now when you're choosing an Aurora Series column, there is no compromise for your proteomics application. So what are our columns capable of? Our 25 centimeter column is our foundation model and the best choice for a broad range of proteomics applications. Here, I present some results for a 200 nanogram heliotriptic peptide analysis. We were able to identify over 7,730 unique proteins from a single shot sample, and this is analyzed on a timsTOF Pro using DiaPassive and the new Diane software suite. We're getting close to 100,000 unique peptides. We're achieving this with spectacular peak capacity with peaks less than 5 seconds at a full width to half maximum, even on a 90-minute gradient. And these high protein identifications are achieved without compromising quantitative accuracy. In an experiment where we mix a yeast digest into a human digest at either a 1:1 or a 3:1 ratio, we observe that a majority of proteins in the mix are accurately quantitated, while also cumulatively identifying more than 10,000 unique proteins across both species in each run. But while identifications from digest standards are very useful benchmarks, what really matters is the impact that our columns are able to have in the research community. One area of particular interest for us is single-cell proteome analysis. And one of the pioneers and the leaders in this field is Associate Professor Nikolai Slavov from Northeastern University. His focus is on developing single-cell analysis methods and techniques that are not only powerful at answering biological questions, but can also really easily be adopted into the community. And here we'll hear him talk about work in his laboratory and also some of his recent achievements.
I'm an Allen Distinguished Investigator and Associate Professor at Northeastern University in Boston, US. My laboratory is broadly interested in systems biology and in understanding the biology of the proteome, increasingly with single-cell resolution. We have lately focused on studying mechanisms of protein synthesis control, as well as protein interactions and remodeling of the proteome at single-cell resolution. I'm quite excited about our recent success in using intelligent data acquisition to increase the number of quantitative protein data points by a factor of 2 using prioritized single-cell proteomics. We are also very excited about the biological inferences that we can make with these data about the remodeling of the proteomes of macrophages as they respond to inflammatory signals. I'm also very excited by PlexDIA, our recently introduced method for multiplex data-independent acquisition, which demonstrates the ability to multiplicatively increase throughput while preserving the quantitative accuracy and depth of coverage that we have known for a while with label-free DIA. And I'm particularly enthusiastic about the prospects of these approaches for single-cell proteomics for reasons that they recently outlined in our JPR perspective. We use both Orbitrap systems and timsTOF instruments. We started using IonOpticks columns in the spring of 2019. We had tried a lot of different commercial columns, some of which performed beautifully and gave very, very deep, high throughput coverage of pristine, highly cleaned up samples, but they were very unreliable and they couldn't be used with samples that weren't cleaned up. And other columns simply performed very poorly in terms of both performance and reliability. And the way IonOpticks columns really stood out for us is by ticking the marks, not in one area, not just in terms of performance or throughput or robustness, but in all of them. They had high enough performance for us, they were very robust, and we liked that this is a technology that everybody can use.
Thanks, Nikolai, for that great insight into your laboratory. Now, another area of research in recent years that have been of great interest to the community is fighting COVID-19. Our next speaker, Alex Campos from Sanford Burnham Prebys, will describe developing a comprehensive SARS-CoV-2 interactome and talk about some of the unique methods he used for distributing those results to the community.
My name is Alex Campos. I'm Director of the Proteomics Core and Associate Member of the NCI Designated Cancer Center at the Sanford Burnham Prebys Medical Discovery Institute in San Diego, California. My main research focuses in proteomics and its applications in the biomedical field. As a proteomics laboratory in one of the hot spots of biomedical research, we have had the opportunity to work with many different groups. I would say that we have focused in different areas of proteomics, including biomarker development, global proteome profiling, drug mechanism of action, and target convolution. And also we have a strong background in protein-protein interaction using mass spec. One of my favorite recent projects is the one that we run in collaboration with Kyle Rhodes. Kyle is the developer of the BioID method, a method to study protein-protein interaction using proximity-dependent ligase. Kyle is also a long-standing collaborator, and last summer he called and said that he wanted to make a contribution to the SARS-CoV-2 research by generating the BioID interactome of the SARS-CoV-2 proteins and the host proteins. So Kyle generated cell lines, A549 cell lines, expressing each one of the SARS-CoV-2 proteins. So he sent the cells to us. We use our favorite workflow to prepare BioID samples, and we can get all the samples prepared in automated fashion. And we inject that in our Orbitrap Fusion Lumos, which is connected to a HPLC hosting with a IonOpticks column, a 25 centimeter column. So we run the whole project, which was the BioID interactome and the global proteome profiling, in a little bit more than 2 weeks. And we thank actually IonOpticks for that, because the data was very robust, very reproducible throughout all those runs. We created a R-Shiny application with all the data there in a way that it's very interactive. You can redo some of the analysis by changing the thresholds, changing the settings, and we do a functional analysis, drug reproposing, and so on. I think that's really nice because most of the typical BioID data set takes about 1 to 3 years to get published. People have to go through the data, mine the data, validate, and so on. So we tried to make this available to the scientific community, specifically to the COVID research community, as soon as possible, and in a way that people can interact with the data in a better way than just a manuscript. We have a total of 5 mass spectrometers. 3 of them run bottom-up workflow. Usually we use TMT or label-free by DIA or DDA. We first learned about the IonOpticks column in 2018. Back then we were using a different vendor, and we started having some problems with robustness and batch-to-batch variation. So a friend of mine told me about the IonOpticks column, and we decided to give it a shot. So since then, we moved all the mass specs to the IonOpticks Aurora 25 centimeter column, and have been pretty happy about it. Has been very robust, very sensitive, and the batch-to-batch similarity is really great. We get very similar numbers. We run actually quality controls pretty much every day. So we inject a yeast digest that we have in all mass specs, and the numbers are very consistent.
Thanks, Alex, for that amazing contribution to the COVID-19 research community. So in addition to deep and sensitive proteome analysis applications shown here by Nikolai and Alex, IonOpticks also offers a range of Aurora Series columns that are used in more high throughput applications. Here I'm presenting data from our 15 centimeter column, where we're able to identify more than 6,000 unique proteins at 50 samples per day. That's using a 20-minute gradient. Now they're really incredible numbers to be achieving in such a short gradient. And in addition to our 15 centimeter columns, we also offer a 5 centimeter capillary flow Aurora Series column for ultra high throughput analysis using a 5 to 10 minute gradient methods. Here we're achieving up to 180 samples per day throughput. Using a 5-minute gradient, we're able to identify more than 4,000 unique proteins per sample from a 40 nanogram HeLa injection with average peak widths below 2 seconds. So what can you achieve with this incredible throughput? Our next speaker, Associate Professor Andrew Webb, who is a proteomics facility head at the Walter and Eliza Hall Institute and also one of the co-founders of IonOpticks, describes how his research laboratory is using our 5 centimeter Aurora Series column to analyze large cohorts of plasma and urine samples from patients.
I'm Andrew Webb. I'm an Associate Professor and Head of Proteomics here at the Walter and Eliza Hall Institute. I'm also a co-founder of IonOpticks, where the original technology that was developed here in my laboratory because we needed a really robust and sensitive chromatography for our LCMS instrument. Over the past 10 years, I've established the Proteomics Lab as an integral part of the collaborative resource with a specific aim of applying next generation research to important biological questions that are relevant to human health. In 2019, I also established a new joint centre, the Colonial Foundation Healthy Aging Centre, which operates between our institute and our local public hospital pathology unit at the Royal Melbourne Hospital. This new centre has a focus not only in making discoveries, but translating them into clinical use as IVDs or in vitro diagnostics. I lead a team of researchers to develop new techniques to advance both basic science and clinically based research. The proteomics team that I lead is directly engaged in the conception, design, and implementation of collaborators' projects and the application and further improvement of cutting-edge proteomics techniques that will drive and facilitate new discoveries in biology. Our current research involves developing both accessible wet and dry lab tools with a focus on automation to further improve proteomics research, and in doing so, drastically increase throughput, as well as lowering the barriers of entry for other medical researchers. More recently, with the aid of a $15 million grant from the Colonial Foundation, we've begun adapting proteomics technology to ultra-large cohorts for the purpose of identifying and translating dementia biomarkers. Our current main project at the Colonial Centre incorporates 2 time points of matched urine and plasma samples from over 12,000 volunteers. These 24,000 proteomic samples of urine and plasma each will be acquired as a single multi-batch experiment. These samples come from a large biobank of healthy aged people that have been extensively clinically followed for up to 10 years. We now know that over 1,000 of these volunteers have been diagnosed with dementia. It's our goal to identify the signatures that will allow us to predict the dementia onset before the onset of symptoms. For the Colonial project, we've had to custom build our own LC hardware to increase the robustness, which now matches the reliability that we see with the timsTOF Pro mass spectrometers. We've also optimized our system around the relatively new microflow column at IonOpticks. This is a 150 micron, 5 centimeter column that has provided us with some pretty amazing results. We've now optimized a gradient of 8 minutes in an 11-minute cycle time. And from this very short gradient with peak widths of around 1.4 seconds, it's enabled us to quantitate 15,000 peptides from 1,800 proteins from most urine samples. We think this is actually going to be quite transformative for urine-based diagnostic development.
Thanks, Andrew, for that great insight into the work in your laboratory. So there's clearly an incredible amount of cutting-edge research being performed with our Aurora columns. We're so proud to be allowing people to unlock the full potential of the mass specs in their lab. Stay tuned to emails and socials because we've got a couple of exciting new product announcements over the coming weeks to months. With that said, I thought I'd leave the final word to those at the coalface really driving the research community forward. I asked our panel of scientists what future trends in proteomics really excite them and why. And the answers they give, I think we've got some pretty exciting times ahead.
I think one of the really exciting things in proteomics is that we are coming to a point when the technology is robust and widespread enough so that there is a potential, still not widely fulfilled, but there is clearly the potential that we can start generating large, highly quantitative data sets from well-designed biological experiments to answer long-standing questions that have remained elusive for a long time. For example, we understand surprisingly little about the protein networks that transmit signal in our cells. Most of the conversation about genetic networks has to do with a very limited regulatory step of transcription factors, binding promoters, regulating transcription, while all of the other regulatory processes involving protein degradation, post-translational modifications, localization, cleavage, all of these processes are poorly characterized, mostly because the technology wasn't up to the level to tackle these questions. And now that we're getting there with robust, high-throughput technologies, I think it's incredibly exciting to think how to marry that technological prowess, that capability of the technology, to well-designed, well-posed biological questions, because we need both to be successful. It's not enough to simply have the technology. It's not enough to simply have the questions. We really have to integrate the two and also integrate them with very powerful computational analysis, which are required to make sure that what we learned from the data is correct rather than artifacts.
The future of proteomics is fantastic. There are many promising technologies out there. I guess a lot of people will mention single-cell proteomics, chemoproteomics, or protein-protein interaction. Although I'm very excited about all these technologies, I want to voice my excitement about the biofluid proteome. I think the revival of plasma proteomics in other biofluids. So our lab established a collaboration with SEER technology, a nanoparticle-based preparation of samples, and we have analyzed some plasma samples and seeing these amazing coverage of the proteins. We are getting an identification of about 3,000 proteins with a throughput of about 8 samples a day. So I'm very excited about this technology, and I hope to see more of that soon.
So there are two main trends that excite me the most in the proteomics field. Firstly is the maturation of ultra-high throughput proteomics, where we can analyse extremely large patient cohorts that will allow us to compare signatures, not just across disease states, but will allow us to truly understand disease in the context of health. Secondly is the ability to perform ultra-deep proteomics, where we can probe the proteome to a depth that includes all detectable post-translation modifications, and where we can do away with any requirement for sample enrichment for these modifications. And ultimately, I think it's the combination of these two things that will be transformational for human health.